through C7, but do a couple of them have a name? What am I talking about?
Atlas.
Okay, which one's the atlas?
It's the first.
Okay. So C1, and does another one have a name?
Axis.
And which one is that? Okay. So if we take a look at these two, we'll see some facets. So the dens of the axis articulates with the fovea dentis of the atlas. Remember, that's just a cool way to write joints. And this forms the atlanto axial joint. This is the no joint. I usually shake my head that way and write the word yes, or the other way around. Then if we look, a similar thing, it'll bring another condyle into the equation, but keep talking about facets. And you see what I mean? The word facet isn't here. Now this is kind of another word for facet. But if we look at the occipital condyles, and we don't need to say it, but we will because we're learning. Of the occipital bone, and they articulate with the superior articulating surfaces of the atlas, and that forms the atlanto-occipital joint. I guess the other thing that comes to mind, at least for me, as we go through some of these words and some of these joints, is perhaps the importance of writing it out as opposed to just looking at it in the bone chart. I know that psychologists that study memory will say that on average we retain about 50% of what we write. So instead of just sitting there looking at it, perhaps looking at it, closing your eyes, trying to recite it, I think that writing it out might be of benefit. All right, let's take a look at these bone parts. Which one did I talk about first? Atlanto-axial. So this just shows typical vertebrae. On the left, you have the sacrum and the coccyx. They're connected to each other in this picture. The sacrum, of course, composed of 5 fused sacral vertebrae. Then over here we have the other types of vertebrae, and typical vertebrae. This is a typical cervical. What does the cervical have? That 2 things actually I'm looking for that allows one to differentiate cervical from thoracic or lumbar. Okay. Well, not just transverse processes, but something in the transverse processes, right? The little holes in the transverse processes called transverse foramen, and they're hard to see in this picture, but those little holes are the passageways for the vertebral arteries. What else do most cervical vertebrae have? Well, they have what's called a bifurcated spinous process. This is a typical thoracic. It has a long, skinny spinous process, and this is a typical lumbar. It's got sort of a square spinous process and a very large body. Anyway, this is C1, this is C2. C1 doesn't have a body. C2 has the dens, which goes into the joint we're talking about. The other name for the dens is the, anybody? Odontoid process. The dens or the odontoid process. So that first joint, the dens of the axis articulating with the fovea dentis of the atlas forming the atlantoaxial joint. Well, that's a facet right there. That's what the fovea dentis is. And on the anterior side of the dens, you kind of have to go out next door into the other room and point at the side of this that we don't see. There's a facet there. And so you can see how that would articulate with that and allow this rotational movement at your neck. Then the occipital, and I think I may have a picture of those connected to each other as well. There's just kind of a close-up of the atlas. The atlas and the axis. And that's how they look when they articulate and allow this movement. The other example we're looking at, and this is the, what is this?
Yes.
Well, this is yes, right? Okay. It's funny the things you forget. All this crap running around in my brain, and I can't even keep straight whether this is yes or no. Just use words. Yes, no. So the other joint up there, if we take a look at the atlas, these are the superior articular surfaces of the atlas. They are facets, and they articulate with the occipital condyles of the occipital bone. Which are shown here in the articulated skull, and here in the disarticulated skull. Now this particular picture reminds me, when you guys are studying things, make sure you study articulated and disarticulated. Don't just study a skull. I pointed out last week we've got a plethora of disarticulated bones, and we have several black boxes that contain completely disarticulated skulls. And you want to study every single bone in there, the itty bitty nasal bones, the little lacrimal bones, the palmar bone, all of them. And I just have to assume you spent the weekend doing that with pictures, because the practical is good. When is the practical? Next week? Week after next? Okay. I wouldn't put this stuff off to the night before, guys. And then finally, a picture like this gives you an idea of how that joint works. As well as this one. I think the hands-on stuff is most important with the tiny bones. The bigger bones, it's relatively easy to get a handle on things with pictures. But when you get into some of these smaller bones, I may not have the right file. Let me try this. Yeah, when you get into some of these smaller bones, like the nasal bones, the lacrimal bones, the zygomatic bones, holding that in your hand and knowing whether it's the left one or the right one, that's a little tough to do with pictures, but it's very easy to do in the lab. We you have the bone in your hand, you have the skull on the table, and you match it up, match the bone up to the skull. So make sure you spend a lot of time doing that. All right, our next category, these are non-articular surfaces. This is letter B under Roman numeral 5 in my notes. So we have surface features that, for the most part, deal with muscle or ligament, muscle or tendon, or ligament attachments. And we're looking at things like a tubercle, a trochanter, a tuberosity, and epicondyle. So you find these, for the most part, on the bones of the appendicular skeleton. You have, for example, with tubercles, greater and lesser tubercles of the humerus. They're up here on the proximal aspect of the humerus where muscles attach. Of course, when you say muscles attach, is that technically correct? How do muscles attach to bones? Histology, tendons. So, we could actually cross that out. Trochanters, you have greater and lesser trochanters of the femur. They're up here on the proximal part of the femur. The trochanters are big things. They look like meat hooks, and that's kind of what they are if you think about it. If they're attachment sites for muscle, muscle is meat. We just generally don't eat human meat. Generally don't. Tuberosities, well, you've got a deltoid tuberosity on the humerus. You've got a radial tuberosity on the radius. You've got an ischial tuberosity on the ischium. And they all look very, very similar. They're slightly raised, they're rough surfaces. And that's really the difference when you compare these three. The trochanter is a big old hook-looking thing. The tubercle is a small, smooth, raised bump. The tuberosity is a rough surface. Then the epicondyles, bring that down here. Those are typically for ligaments. So again, histology, tendons, muscle to bone, ligaments, bone to bone. So the dense regular connective tissue holding bones together generally runs from one ligament to another ligament. These are certainly far easier to see in the in the lab than they are in a picture, but just to give you a couple of ideas. And then with the epicondyles, we'll draw those because that takes us to an important point where the elders who make all this crap up decided to mess with us a little bit. So right up here, proximal aspect of the humerus, that's where your greater and lesser tubercles are, and I could actually point to them, but I don't think you can actually see them, greater and lesser. An interesting tuberosity to use as an example, well, this will actually show you the greater and lesser tubercles of the humerus. There's the greater, there's the lesser. So they're raised, but they're smooth. They're not like big old meat hooks the way the trochanters are. We go down here to a femur. You can get an idea what I mean about raised meat. This is the greater trochanter. That's why I kept giving you this as an example. Looks kind of like a meat hook. But an interesting tuberosity, because, and I bring this up because a nice way to learn a lot of this stuff is to touch yourself, palpate it. You probably don't want to palpate this one in the mall. Might be, you know, people might look at you. You could palpate the deltoid tuberosity. Nobody'd think anything about that. But the ischial tuberosity, you might get some strange looks. So if we use any of, like this picture, just for reference, you can see where it is, posterior, inferior aspect of the os coxa. But then if we go to that os coxa picture that we looked at on Wednesday, you can see it very, very prominently. So again, it's just a rough surface. Okay, as I said, for epicondyles, we'll draw a picture of our own because this takes us to another important point. So if you have a femur, and you have a tibia, and we use the words that we learned last week and have elaborated on this morning, you would certainly call those condyles, and you'd have to say of the femur because there's a ton of condyles, and you'd have to say left and right. So if we give this a little perspective, this is anterior, so this is anterior. If we're looking at that leg, so is that the right or the left leg?
Right.
It's the right leg. So this would specifically be the right lateral condyle of the femur versus the right medial condyle of the femur. But if we use the language, the terms, the parts, the words from last week and that we've elaborated on this morning, what would you call these things? I know what you'd call them. You're just asleep or something. You'd call them fossa. But years ago, somebody decided to play a trick on us. They're condyles. Yeah, they're condyles. They don't look like condyles, they look like fossa. It's kind of mean to do that to us, but it's the truth. It's the truth. So the right lateral condyle of the femur articulates with the right lateral condyle of the tibia. Well, that's easy enough, but why is it a condyle and not a fossa? I don't know. I didn't get to make up the name. I'm just here to share it with you. Now, as we continue, we'll put another picture just like that up here. What's epi mean? Above? Exactly. Yeah, epi means above. Okay. And I've said that epicondyles are where ligaments attach to hold bones together. So if we have a condyle, and if we have a ligament holding those bones together, another one over here, so in other words, on each side of your knee, you have ligaments that hold the bones together. Those ligaments are attached to an epicondyle. That's what we're talking about, an epicondyle or epicondyles. So that makes sense. Epi means above. The epicondyle is above the condyle. And we already have the fact that it doesn't make sense to call these things condyles. They look like fossa. But look at the name of this. Why didn't they call that a hypocondyle? I don't know. I didn't get to make up the words. I'm just here to share them with you. It goes from maddening to interesting to downright funny. It goes from difficult to simple. Next semester you will look back on all of this and wish it's all you had to do, think it was a joke. Right now it's overwhelming. I understand that. I appreciate that. It's not any easier because of what seems to be inconsistency. We get our brain wrapped around what a condyle is versus a fossa, and then they're going to call this fossa a condyle. Crazy, absurd.
Are there more exceptions where a fossa is called a condyle other than?
I'm sure there are. I'm sure there are. Don't have one off the top of my head. Let that run, and maybe I'll come up with one. We'll see if I can process that in the background. But in science, overall, it's the exceptions to things that are fascinating and interesting, and God, especially in drug chemistry, it's the exceptions that make it so bizarre. You think you've got a handle on this, and oh my God, didn't do at all what I thought it would do. Didn't do what it should have done. Let's see, are there any, I mean, there are, now, keep in mind, we're looking at examples, okay? Roman numeral 5, letter A, I did not cover all articular surfaces. That's what the bone chart's for. Roman numeral 5, letter B, non-articular surfaces. I didn't cover all of them. There's one more I want to mention. Bizarre word. The word is process. What is a process? Well, any surface feature. So a tubercle is a type of process. A trochanter is a type of process. An epicondyle is a type of process. A head is a type of process. But then the word process is also used another way. So if we have two bones, and This is actually logical. So if we call this bone A, and we call that bone B, then this is the B process of bone A. This is the A process of bone B. Get it written down and then stare at it and let's figure it out. What's it mean? So why is this called the B process? Because it articulates with bone B. So the B process, and now we show possession. Who does the B process belong to? It's the B process of bone A. So it's called the B process because it articulates with bone B. And then the other one works exactly the same way. It's the A process. Why? Because it articulates with bone A. It's the A process of bone B. Now, further maddening, perplexing, these are listed under non-articular surface features. Well, isn't this an articulation? Yes, but it's a certain type of articulation. It's a suture. The joints in your skull, we talked about those last week. You can't wiggle your sutures. It'd be funny if maybe you could. So I'd like a volunteer to come up here and wiggle your right parietal bone. You can wiggle your hand, your wrist, your leg. Come on, somebody, anybody, wiggle your right parietal bone. You can't do that. These are sutures. So, and are they articulations? Yeah. So why aren't they in the category of articular surface? I don't know. I didn't get to make up the rules. They're in the category of non-articular surface features, even though they're articulations. They're just immovable articulations. Now, one thing you do have to wrap your brain around as you start studying articulations, and that won't be today, but it's not terribly far away. When you say articulations or joints, and that's all articulations are, you have to throw out the window what you've thought about as joints all your life. These are freely movable joints. They're one category. But where your teeth, and some of you know, because some of you studied, your teeth are in little sockets called alveoli. Those are joints. Natalie, wiggle your teeth for me. Don't use your finger. Okay, those are joints. So joints aren't just these things. We have lots of joints that are immovable. So let's look at some examples of this nomenclature, and that's what it is. Just like we have nomenclature in chemistry, we have nomenclature in taxonomy. So this, call that X, call that Y. X is the zygomatic process of the maxilla. Of the maxilla, because it belongs to the maxilla. Why is it called the zygomatic process? Articulates with the zygomatic bone. Okay, so now name Y. Maxillary process of the zygomatic, exactly. Okay, this one, and this one's on your bone chart, I know. Put a little Z there. That's the frontal process, because it articulates with the frontal bone, frontal process of the maxilla. If we rotate this thing, so we're looking at the side, you'll see a couple of more that are on your bone chart. So somebody name that one for me. Zygomatic process of the temporal bone. And so this one is the temporal process of the zygomatic bone. Somebody name A for me.
Zygomatic process of the frontal
Zygomatic
bone.
process of the frontal bone, and so B is the
Frontal process.
frontal process of the zygomatic bone. Now you're going to be able to do that when I give you a disarticulated zygomatic bone. And tell me whether it's left or right. Yes, you are. I'm telling you, you've got to be able to do that, okay? I'm telling you, we've got these black boxes with disarticulated skulls. You've got to sit there at the table with a picture like this, or a skull, and the little disarticulated bone, and hold it up here, and figure something out that works for you. Like you might say, well, the widest articular surface, is that the widest? You've got this one, and this one. The widest articular surface articulates with the maxilla, and so you may write that down. And see, the act of writing it down is going to burn it into your brain. And then how could you describe this one, the frontal process of the zygomatic? What might you write down to help you remember it versus the other two? I mean, you've got the widest is maxillary. This one is the
Superior.
but you don't know what's superior. You've got the disarticulated bone. So what'll work?
The process along the orbital.
The narrowness. Long, slender, narrow. Okay, yeah. So the long, slender, narrow is the frontal process of the zygomatic. The widest is the maxillary process of the zygomatic. And so then by default, the other one is. That's how you're going to do it. Yeah. And then when you've got all that, you're going to be able to hold it in your hand, and is that the left or the right? Okay. Remember, please, open lab policy. You guys are welcome to come to lab 6 days a week. 4 201 labs, 2 202 labs. Pictures are great. A lot of people have done it with nothing but pictures. But when it comes to these small disarticulated bones, I think the time in the lab is extremely helpful, extremely valuable, necessary. Maybe I should just put it that way. Okay, finally, our last category, Roman numeral 5, letter C, we have depressions and openings. So one of the biggest ones here, the foramen. So many of those. And it's one of those things where I say, as I've said so many times, it's not difficult. It's just rote memory. So, you've got to know what goes through the jugular foramen. Canal is similar. I'll differentiate between those in a minute. Fissure, similar. I'll differentiate between those in a minute. There's one under the occipital bone. What's that mean? Big hole. The sphenoid bone, you've got foramen rotundum, ovale, lacerum, spinosum. It's not difficult, guys. I'm begging, I'm telling you, you have to know. I'm telling you, I'm going to ask you. I'm going to ask you on the practical, I'm going to ask you on the exam. I'm telling you, you absolutely must commit it to memory. If this was second grade, and I said, I will ask you multiplication table questions on the exam. You have to know 2 times 4 and 6 times 7. You have to know that, okay? Would you go home and memorize them? Yeah. You did. You all did. This is no different. It's not fun, but it's really simple. It's rote memory. Rote memory. I was 47 years old before I knew the word was not R-O-T-E or R-O-P-E. No, I'm kidding, but anyway. All right, so a canal, think about a doorway versus a tunnel. A doorway is like a foramen. There's no appreciable horizontal distance traversed when you go through a doorway. Okay, there's a few inches there, but there's no real, compared to a tunnel. Okay, tunnels can be quite long. They don't have to be, like the Eisenhower Tunnel, but in a tunnel you traverse a significant horizontal distance. In a foramen you don't. So foramen is like a doorway, a canal is like a tunnel. A canal is also called a meatus. We have
Will you spell that?
meatus, M-E-A-T-U-S. We have an interesting situation. When we look at the foramen lacerum, get both of these on the screen at the same time. No, I can't, but you can certainly get your bone charts out. So if you look at the undertemporal bone, the carotid canal, and then go to probably the next page. You've got your bone chart in front of you. You can figure it out. You're probably quite familiar with it by now anyway. Don't have to figure it out. Go to the sphenoid bone. Look at the foramen lacerum. So this blue vessel is the pharyngeal artery. This red vessel is the internal carotid artery. So if you're looking in the skull, okay, at the top, right here, where foramen lacerum is shown, you're going to see two vessels coming up through it. And so when we memorize what goes through the holes, we associate the pharyngeal artery and the internal carotid artery with the foramen lacerum. But when we look from the side or from the bottom, we can see that it's actually only the pharyngeal artery that truly goes all the way through the foramen lacerum. The internal carotid enters the carotid canal. Remember, a canal is like a tunnel, traverses a significant horizontal distance, and then the carotid canal intersects the foramen lacerum, and the internal carotid artery goes up. And from the bottom, it's quite clear that they're two totally separate openings. But from the top, it appears that the two vessels pass through the same thing. So what are you going to do in the lab that you saw me do on the DVD to get your brain wrapped around this? Pipe cleaners. You're going to use pipe cleaners. You're going to get a blue one and a green one, or whatever colors are in there. Yeah, that's what you're going to do. You're going to use pipe cleaners. Now a foramen is a hole with no significant horizontal distance traversed, like a doorway. A canal or a meatus is like a tunnel. Driving in your car, you traverse a significant horizontal distance. Doesn't have to be as significant as the Eisenhower Tunnel, but it's not like a doorway. And then finally, a fissure. How would you describe a fissure versus the canal or the foramen?
Small slit.
A slit. Fissure, your keyword is a slit. I guess we have to say that word carefully. Which we really don't, we have a First Amendment. Okay, so there's the superior orbital fissure. And that's the maddening, when you've heard me regurgitate what goes through it several times ad nausea, the abducens nerve, the trochlear nerve, the oculomotor nerve, the ophthalmic branch of the trigeminal nerve. Yeah, guys, you gotta be able to say that, it's no big deal. But how are you gonna learn to say it? You're going to write it down. You're going to write it down. You're going to take the time to make sure you spell the words correctly? Yes. If you want credit, you will. Correct me if I'm wrong. There's a cranial nerve chart on the Facebook page. Okay, make sure you have that. And it lines up the cranial nerves with the framing they go through. Verification? Okay. Yes?
Yeah.
Okay. Make sure you have that. Okay, make sure you have that. Now, make sure, because I can picture the chart, I don't have it to pull up. So, third time, someone verify for me. It gives you not only the name, like abducens or trochlear, but it gives you the number. Yes? Yes. Make sure that you're listening to me. One of you is sleeping. I'm not going to single you out, but wake up. That when you name a cranial nerve, you give me the name and the number. Make sure you do that. The name and the number. I only say this because I learned that it was wrong of me to assume everybody could count using Roman numerals. Most people can, but some people can't, because we don't do it on an everyday basis. So you must be able to count from 1 to 12 using Roman numerals. Okay, there's no such thing as number 2. Okay, it's just wrong. Okay, so don't give me this.
Sure, did you say there were 12 of them?
There are 12 pairs of cranial nerves. They don't all come up this semester.
Oh.
So your chart may not show them all.
Okay, it's all off the
But
only.
you could very, very easily use the index in your book or Google if you were interested in the 12 names. Absolutely.
Thank you.
But you don't have to, but I mean, it's right there. Yeah, there are 12 of them. Okay, any other depression opening stuff I want to mention? I've kind of already mentioned it, just to remind you though, the sockets that your teeth are in, they are alveoli, just like in the lungs. Alveolus, alveoli, and they are joints. We'll define those joints probably next week, but they're called gomphoses. Another word that's fun to say, or perhaps not. Okay, so a bunch of stuff to make sure you're wrapping your brains around it. This takes us to Roman numeral 6, bone physiology. If you look at the two letters there, letter A, bone development, letter B, bone cells, hormones, and homeostasis, which of those have we talked about?
B.
Homeostasis, exactly. So your bone cells, bone hormones, homeostasis. So as I'm reminding you, you might want to flip back in your notes, find that in your notes. And not that I'm going to tell you guys how to study, I'm going to tell you what I did when I was in this world 76 years ago. Right here, I would say, see October 1st, or see September 23rd, page 4. And I'd write that right here in today's notes. So if you'll flip back in your notes and find where we talked about bone cells, so you've got osteoclast and osteoblast, the hormones, calcitonin, parathyroid. You remember having all that discussion? It was in the context of homeostasis. And homeostasis was in the context of histology because of the pancreas and the islets of Langerhans and the alpha cell. Okay, you remember all that? So, jot something down here now so that when you're home studying, you've got it, and you can get to it when you need to. So the actual bone development, letter A, is what we need to talk about, and that will be the last thing we talk about today, whether that means we get out early or get out late. I'm guessing we'll be pretty much on time. So an easy word to break down: Genesis, formation, creation, osteo, bone. So bone formation, bone development, I think is the word I use in the notes. Buckle your seatbelts. This is a wild story. So as you learned in biology, males are 44XY, and females are 44XX. And when they undergo meiosis, or gametogenesis, or spermatogenesis in his case, oogenesis in her case, so the male forms sperm, they may be 22X, they may be 22Y. The female forms eggs, they have to be 22X. Only sex chromosomes she has are X. So one of the sperm, X-bearing sperm, Y-bearing sperm, it doesn't matter for this story. But one of the sperm fertilizes the egg. And so we get a one-cell embryo. Which is called a zygote. Now we learned in histology where this takes place. And we know that that embryo moves through the fallopian tube to the uterus to implant. And as that embryo is moving through the fallopian tube into the uterus, it is dividing. Etc. Up until somewhere around this point, all of the cells are omnipotent. So any one cell is capable of forming a complete human being and placenta. Twins, of course, identical twins occur when a two-cell embryo fractures and each cell develops into a human being. That's why they're identical. Versus two sperm, each sperm fertilizing one egg, then they're as different as you and your brother or sister who were born, a year apart. Well, we get a division into two groups of cells. Cells that will form the embryo versus cells that will form the placenta. And then we get a division into cells that will form epithelial tissue versus cells that will form connective tissue versus cells that will form muscle tissue versus cells that will form nervous tissue. And you think of all the different connective tissues. You've got your fibrous stuff, adipose, you've got your supporting bone, cartilage, all of that. So we get many, many, many more subdivisions here as this embryo is growing. But the two that matter for us, because this is not an embryology class, we get cells that will form membranes versus cells that will form hyaline cartilage. I mean, it's kind of cool, isn't it, to think that one haploid cell from your dad, called a sperm, and one haploid cell from your mom, called an egg, fused to form one diploid cell, and from that you came about. You're here. And that one cell had to divide into billions and billions of cells. And some of them had to say, I'm going to be your brain. Some of them had to say, I'm going to be your kidney. One of them had to say, I'm going to be your left big toe. Etc., 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 etc. And as far as the ones that said, I'm going to be your brain, you've all had Psych 101. How did the cells that said, I'm going to be your brain, decide, I'm going to be your thalamus? I'm going to be your pons. I'm going to be your medulla. How? I know. Let's look. Somebody text God, somebody text Darwin. Let's see who gives us an answer. Because I sure don't know. So the connective tissue cells, you have some that form membrane, and there's some that form, it's too low, I'm going to run out of room. The cells that form membrane take a path called intramembranous ossification. And the cells that and through intramembranous ossification, they form bone. These connective tissue cells that become hyaline cartilage take a pathway called endochondral ossification. And become bone. And you can look at your notes, numbers 1 and 2 under letter A, you'll see the terms intramembranous versus endochondral ossification. Draw from histology, number 1, you know that in the world of connective tissue, you have fibrous connective tissue, you have supporting blood, fat, all the other stuff. Draw from histology, location of hyaline cartilage, fetal skeleton. So from this one cell embryo called a zygote, after it divides somewhere between 64 and 128 cells, we get this initial differentiation into cells that will become the placenta versus the embryo. Why did I point at embryo when I said placenta? And then within the embryo, we get the cells that will form the 4 primary groups of tissue. And then we sort of just focus now on what we're talking about. We don't get into all of them. Within the world of connective tissue, we have cells that will form membranes versus hyaline cartilage. And then we have the 2 mechanisms of making bone. The bone made from membranes, this is largely how we make flat bones versus the bones made from hyaline cartilage. This is how we make long bones. Now you have to appreciate how superficial this is, because in addition to flat bones and long bones, we have short bones, we have irregular bones. So it's like everything we talk about, guys. You could take an entire class called osteogenesis. It would be a graduate school class, but it's out there if you were interested in this. So what we need to look at now are these 2 mechanisms of forming bone. We'll look at the endochondral mechanism first. So you pick a long bone to imagine. Maybe you're imagining the femur, you're imagining the ulna, it doesn't matter. The bone, the epiphyses are going to have articular cartilage. This from Wednesday, Anatomy of a Long Bone. The bone is going to be covered by a membrane called periosteum. I failed to mention that on Wednesday, but you picked it up when you read the book. And so what we have here is a little cartilaginous model of the bone. Again, you can picture the femur, the ulna, the radius, the humerus. It doesn't matter. You don't all have to picture the same bone. And it's tiny. You need a microscope to see it. But it looks just like the femur or whatever bone you're picturing. In fact, you'd be looking at this embryo under a dissecting microscope. You use those in Biology 111. They're the microscopes that only magnify up to either 10 or 100 times light. And they let you see 3 dimensions. And you probably looked at pine cones or your creases in your fingers. I don't know what you looked at. A stamp, maybe. So the whole embryo is tiny, tiny, tiny. And so one bone is super, super tiny. Well, the first thing to happen here is a blood vessel penetrates the perichondrium. The perichondrium is just the same thing as periosteum. Peri is to cover. So if we say periosteum, we're saying cover the bone. But right now the bone isn't bone, is it? The bone is what? Hyaline cartilage. So it's called perichondrium instead of periosteum. It's the same thing. But we can't say it covers the bone when the bone isn't bone. The bone is cartilage. So the blood vessel grows through the perichondrium. So there it is. The cartilage cells swell and they explode. Osteoblast. absorb calcium. So right there in the, what part of the bone is that? Diaphysis. Right there in the diaphysis, ossification is occurring. So osteoblasts absorb calcium in the diaphysis. But you knew it was the diaphysis. You can see the blood vessel is not in the epiphysis, it's in the diaphysis. This establishes what is called the primary ossification center. So your little cartilaginous femur is becoming bone. The ossification begins right in the middle of the shaft. Stated more correctly, the diaphysis. This process is repeated in each epiphysis. So here we go, blood vessels, bang, bang. The cartilage cells swell, explode. Osteoblasts begin to absorb calcium in the, what are you going to change this word to? Epiphyses, because it's happening at both ends. So spell it ES instead of IS. And so what are you going to call these centers? Secondary ossification centers. So now your little cartilaginous femur is becoming bone right in the center of the shaft and at each end. Ossification is going to spread. Okay, so from each end, ossification spreading toward the center. From the center, ossification is spreading toward each end. Now you're bored now. In fact, you're probably snowboarding. And you end up with the entire bone ossified, except one spot on the proximal end and one spot on the distal end. Lay people call these the growth plates. So that little cartilaginous femur has ossified except for one place on each end. That growth plate is still hyaline cartilage. Ossification continues anywhere from 12 to 20 years after birth. some cases, a female doesn't elongate anymore after 12 or 13 years old. In other cases, a male grows another couple of inches between ages 18 and 20. So we can't put an exact time here. But, you know, somewhere 16, 17, 18 years old would be the most common range. You are through elongating. Oh, you're still going to grow, but you're through elongating. What's the technical term for that growth plate? Anybody encountered that?
Is that the epiphyseal line?
Yes. It certainly is. And then it becomes what? Actually, hang on. Epiphyseal plate. And then it becomes the epiphyseal line. When growth is complete. So now if we took the femur of a 24-year-old, there wouldn't be any hyaline cartilage here, but we'd still be able to see where it used to be. But if we took the femur of a 64-year-old, we wouldn't even be able to see it. So it's a method that a coroner could use to estimate the age of death. Just like, and I don't have a picture of that, but just like looking at any of these sutures in the skull. The older you are, the more ossified those sutures become. So you find a skeleton in the desert, the coroner can figure out, is this a 20-year-old, a 40-year-old, a 72-year-old, an 85-year-old, by looking at the degree of ossification in these sutures. You can do the same thing looking at that growth plate. Now, speaking of growth plate, imagine a fracture through the growth plate. So now one end of the bone may not elongate at the same rate as the other end of the bone, or the proximal right femur, the proximal aspect of the right femur may not elongate at the same rate as the proximal aspect of the left femur. So you can have, an arm length discrepancy, a leg length discrepancy. So guys, this is your, back to histology, location of hyaline cartilage, fetal skeleton. And why do we need this? Well, you think about, and again, we started with an almost microscopic embryo, certainly microscopic bone within the embryo. We went out to 18 years old or so, but why do we need this? Well, think about all the things that kids have to learn to do. I mean, do they fall a lot when they learn to walk? Okay, so what's good that their bones are flexible. Now, if you go home and do this to your child's femur, I'll give you an F in the class, which you could if your child's, a little it. What else do they do when they're little kids? Don't they get in trouble? Don't they crawl up on the counter? Sometimes they fall off. They try to crawl out of their crib, crawl out of their plate, and don't they fall? Don't they fall? Don't they fall? How do you protect them? Well, you don't. You don't have to. God or Darwin already did, giving them cartilaginous bones. They learn to ride bikes. They learn to snow ski, snowboard, hopefully both. In today's world, man, it's hard. I was 40 years old before I ever snowboarded. That hurts. That's painful, painful, painful. I learned to ski when I was 2 at Sun Valley in Idaho, and I'm probably as comfortable on skis as I am walking. I'll never be comfortable on a snowboard. Never, 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 ever. All right, we ran out of time, so we'll deal with intramembranous, any pen, please. We'll deal with intramembranous on Wednesday. Thank you. Is your, I don't know if you've had a chance to look, but did you get your exam grade? Okay, is it MD2L? Okay.
Yeah.
So is the practical one.